Okay, so we're going to start the flash review for DNA and protein synthesis. Some of the really important things that you need to know for your upcoming assessment. First off is going to be DNA replication. Remember, DNA replication means making a copy of the DNA. So to start with, DNA replication follows what we call a semi-conservative model. And that's really important vocabulary for you to look for in answers. So semi-conservative model. Semi-conservative means that the DNA is half saved. And if we look at the picture of the blue DNA, we can see what that means. We start with the original double-stranded DNA. It gets separated, it gets pulled apart, and then each one of the original strands is used as a template or a model to build the new strands. At the end, we now have two double-stranded DNA, each containing one original strand and one newly built strand. So it is half saved from the original, semi-conservative. That's where that wording comes from. Always important to remember that DNA replication occurs during the S phase of the cell cycle, right there during interphase. And when you think about DNA replication, the thing you have to do is to be able to describe how this process conserves and transmits DNA to the next generation. And think about it for a second, and the answer should pop right up for you. This process conserves and transmits DNA to the next generation because, since DNA is double-stranded, one of each of the original strands is passed to the next generation. So it is saved and passed. Okay? That's what you're looking for. Each DNA molecule, when it is replicated, consists of one original strand and one newly built strand. Next thing we want to work on is the central dogma of biology, or how cells make proteins, protein synthesis. Now this is important because this is how all cells make proteins. Whether you're a bacteria or a killer whale, you make proteins the same way. The information in DNA is used to make RNA, is used to make proteins. It's a two-step process, okay? And that first step is called transcription, where the information in DNA is used to make RNA. The second step is called translation. Notice transcription and translation are in alphabetical order, so you always know which one goes first. Transcription makes RNA, translation makes proteins. Okay? Now, the other thing about this is, where do these processes occur in a cell? Well, transcription involves DNA, and DNA is always located in the nucleus, so transcription occurs in the nucleus. Translation is when that information in the RNA is taken to a ribosome. So translation occurs at a ribosome. Those are located in the cytoplasm. You know translation occurs at the ribosome or in the cytoplasm because ribosomes make proteins and translation makes proteins. So you want to think about this in the big picture. What you're looking at right now answers half of the questions that you could have on protein synthesis or central dogma. So just think about the picture and think about how things fit together. So let's answer a couple questions. The picture that you see on the right-hand side shows you the same thing that you saw in the last picture. It's just angled differently. So DNA is being copied to make RNA, and then you have another arrow down that points to a polypeptide. So transcription is the beginning here, where you start with the DNA and you use that to make the red line. So the first question, A, what is the molecule being produced by transcription? You can see it written in the picture. It says RNA. RNA is being built using DNA as a template. That's transcription. Question B is asking you what's the name of the process that's making the polypeptides or proteins. And we know that this is a two-step process, transcription followed by translation. Awesome. So both answers are there. Okay, so this process, you'll get used to seeing it in different ways, but it's the same. Transcription followed by translation, and you know exactly where they occur.